Hello friends, good day to all. Today we have come with another important new topic, that is the spermatogenesis. This is the second part of male reproductive system. So before entering this topic, I would highly recommend you to please go and watch my previous video part 1 of male reproductive system so that you can understand better. So with this, let us proceed. My dear friends, whenever we come across any words, the easy and simple way of learning this is to split these words and separately take the meaning. So here, what is the meaning of genesis? Here, genesis means the formation, the process of formation. So at the same time, this word is derived from the sperm. Sperm is the male gamete. So now you have combined this meaning. It is that the process of formation of male gamete, that is sperm. So it is very easy to understand like this. So my dear friends, the first question arises here is, what is the site of spermatogenesis and what is the source from these sperms are being produced? My dear friends, if you have gone through my previous video, I have very clearly made you understand the concept of spermatogenesis, that is arising from the testis. So testis, the site is actually, we have discussed, the organ is the reproductive organ, that is a primary sexual organ. Because this is the primary sexual organs, the characteristic feature is that they are only gamete producing organs. So in case of males, it is the testis. But to be specific, we also have learned that this spermatogenesis is occurring in the seminiferous tubules. So now we are going to learn how in these seminiferous tubules these spermatogenesis is occurring. So now the question arises, what is the source in the seminiferous tubule from which these spermatozoa are being formed? The answer is very simple, that is we have already learned, that is the spermatogonium. This is actually, all these sperms are being produced basically from this by many cell division process. Different cell divisions can be mitosis or meiosis and leading to the formation of sperm. So this is also known as the spermatogonium mother cells or spermatogonium stem cells. So this is how we have to correlate and learn the nomenclature as well. So my dear friends, let us go now into the actual what is the flow chart, and so that we can understand better. So my dear friends, as you all are seeing here, this is the seminiferous tubule. And so if you take the section of seminiferous tubule and enlarge, so this is how it is. So the inner part, this is the lumen, and this is all the lining epithelium, and this is all the lining epithelium. And here there are two types of cells in the seminiferous tubules generally, and in relation to the seminiferous tubule there are three types of cells, but here there are two types of cells, because here generally it is lined by what? Seminiferous tubule is lined by germinal epithelial tissue, and this germinal epithelial tissue will be having spermatogonium. So this is all how it is arranged. So this will be having the spermatogonium cells, and this is lined by the germinal epithelium, and germinal epithelium will having spermatogonial cells. And to be specific, there are some another Sertoli cells as well, as Sertoli cells. They are helping cells, or the nerd cells, and we'll be discussing in detail next. So here in the context, let us proceed how the spermatogonial cells are forming the spermatozoa. So here, as you are seeing, the resting spermatogonium cells will undergo many cycles of mitosis, cell division, and form the type A and type B spermatogonium cells. And what is important to note here is that type A cells will be reverted back in the cycle, whereas only type B cells are proceeding forward in the spermatocytosis. So here the type B cells are undergoing another mitosis division and forming the primary spermatocyte. Up to here only mitosis division is taking place, but from the primary spermatocyte the meiosis division starts, because here the primary spermatocyte undergo meiosis 1 and form the secondary spermatocyte. As we all know, if you have at least a bit of knowledge about the difference between the meiosis and mitosis, you will understand that this meiosis is known as the reduction type of division. That is the reason why here the chromosome number is reduced to half. So it has formed a secondary spermatocyte, and this secondary spermatocyte undergo meiosis division 2 and lead to the formation of spermatids. So my dear friends, now next what is happening is you have to analyze here that in the next process the number of spermatids are not increasing. So here are 4 and here also 4, so both are number is 4. Here when you see it is 1 and forming 2, so here it is 1 and forming 2. So all these processes are division process, whereas here as the number count is same in both, so there is no cell division here. Whereas then the question arises, what is happening that is making the spermatids to spermatozoa, and what is the special name for this process under the spermatogenesis? It is also a part of spermatogenesis, but we have named a special name for this process because here there is no division, as well as there is also a changes in the form of the cells. So these changes of form in the cell is known as metamorphosis. So the process of metamorphosis that is leading spermatids to spermatozoa, this is known as the Spermiogenesis. Now let us move to our next slide so that we can understand more in a better process. Before that, I would like to stress one point that here as you are seeing the lining epithelium and is giving the spermatogonium, and this slowly as there is a division taking place, they are moving towards the lumen, so that at the end of the process these spermatozoa formed can be easily released into the lumen. And for your kind information, the releasing of mature spermatozoa into the lumen is known as spermiation. This is very important. This is spermiogenesis and this is spermiation. So this is all the terminology. Generally students get confused, but I will explain in detail in another slide as well. So don't confuse. For this slide, you just remember that these cells are, as they are dividing by any means, mitosis or meiosis, they are moving towards the lumen so that they can be released into the lumen very easily at the end. So after releasing into the lumen, how they are passed outside the body via different ducts, I have clearly given all the idea and the concept in my previous class of anatomy of testes. Please go and watch. So let us move to our next slide. So my dear friends, in this slide you can see very clearly here that how we have differentiated meiosis, spermiogenesis and spermatocytosis. My dear friends, this 1, 2 and 3, all these are part of spermatogenesis only. All these three come under these are spermatogenesis. So all these are just the parts of spermatogenesis only, so don't get confused. So here in the spermatogenesis only, up to the primary spermatocytes, as there is only mitosis taking place, so this is named separately as spermatocytosis. Whereas this primary spermatocyte to conversion of secondary and secondary to spermatid, all this is under the meiosis. And third stage is the metamorphosis state, that is from spermatids to the spermatozoa. This is Spermiogenesis. Whereas the release of spermatozoa into the lumen, that process is known as spermiation. That process is known as spermiation. Now I would like to bring to your notice that how these number of chromosomes are being reduced and changed along the process, and I will also discuss what is the difference between small and capital N. So if you write small n and if you write capital N, what is the difference? So here small n is changing. So my dear friends, spermatogonium also has 2n, all these has 2n, and up to primary spermatocyte all has 2n, and as soon as this meiosis 1 takes place, the number is reduced to half, so n, all these are n. So here what we have to note that due to this meiosis 1, that is the reduction division what we call, so the sperm count has been halved, whereas here in meiosis 2 the sperm count has not halved. Why? Because if you go in depth and learn what is meiosis, you will definitely put one point that meiosis 2 is similar to mitosis. That is the reason why there is no reduction of chromosome number. So then what is the meaning? What does indicate capital N? Capital N indicates just the amount of DNA in the cell. So this is the important topic usually asked in MCQs, and also many students get confused and neglect these points. So please don't do that. So my dear friends, we have completed this all the things. Let us move to the next and try to learn the spermiogenesis process in depth. So as we all know that in the process of spermiogenesis, that is a part of spermatogenesis only. In the spermiogenesis, there is no cell division occurring either in neither mitosis nor meiosis. So only metamorphosis is occurring, and in this metamorphosis the spermatids are transformed by changing in the form, by bringing changes in the form from spermatid to spermatozoa. So what are all those forms? What are changes taking place in its form, shape or form? So let us see all those things. So here you can see very clearly that this spermat is how it is transformed into mature spermatozoa, how these Golgi apparatus and this nucleus, these centrioles and the mitochondrions are being changed into another forms. So first, if you see, this Golgi apparatus is changed into the acrosomal cap, that is covering two-thirds of the nucleus anteriorly. So and this nucleus has condensed and moved towards anterior pole. So how this nucleus has been condensed and moved towards anterior pole. At the same time, you can also appreciate this centrosome is divided into proximal and the distal centrioles, and proximal is forming the axial filament, and that is lying in the neck region, whereas the distal will be present here in the middle, at the junction of the middle piece and the tail. So apart from this, you can also see here that how mitochondria has developed into a spiral sheet that is covering the encircling around the axial filaments to form the middle piece of the spermatozoa. Whereas we also have to know one more thing, that the cytoplasm has shaded into a residual body known as the Raynaud's residual body, known as a Raynaud's residual body. All these 1, 2, 3, 4 and 5, all these are the changes that are occurring during the process of spermiogenesis. And this is all the changing from the metamorphosis of spermatids to spermatozoon, whereas the release of spermatozoa into the lumen is known as the process of spermiation. You have to differentiate these two processes and do not get confused. After knowing all this, if you study in a conceptual manner, definitely you are not going to confuse. So with this, let us move to our next slide. So my dear friends, After all these things, after all knowing the how the process of spermatogenesis is occurring and how the ploidity is changed all throughout the process, and the chromosome number and the types of divisions and so on. So now the question arises, when does this process of spermatogenesis starts in the male? Either it starts before the birth in the fetal embryonic period, or it starts or just after the birth during the childhood, or it starts at the age of puberty. The answer to this question is, the answer is at the age of puberty. What is reproductive period? So reproductive period, it is the period in which the formation of gametes take place only in this reproductive period, and the period starts with age of puberty, that is nearly 10 to 14 years. So it starts with the age of puberty, but the ending of this period differs with sex. So in males it can last much longer than females, whereas in males it is longer, whereas in females it extends up to 45 to 50 years only, up to 45 to 50 years only. After that the menopause occurs. Here the menopause occurs. So from puberty to the menopause, whereas in males from puberty to longer than the period is extended beyond the 50 years as well, so depending on the productivity. So after knowing these things, the question arises, what happens at the age of puberty that is stimulating this process of spermatogenesis? That is very important. What is happening that is stimulating? What is the stimulant? What is the thing that is leading to the initiation of this spermatogenesis? So in order to understand this, let us focus on this chart. So here you can see that as soon as the onset of puberty, in response to the onset of puberty, you can see that hypothalamus that is lying here, this hypothalamus releases one hormone, what is known as a gonadotropin, releasing hormone. So this gonadotropin releasing hormone has positive effect on the anterior pituitary, and this anterior pituitary, what it will release another two special hormones known as, one is the luteinizing hormone, another is the follicular stimulating hormone. One is LH, another is FSH. Whereas one important thing is follicular stimulating hormone. Here, what is the function of follicular stimulating hormone in males? What is the function of follicular stimulating hormone in males? Because follicles are present in females, right? Whereas though the follicles are present in the female, this same hormone is also found in males. But what is the function is very important. The questions might be asked in any ways, so you have to be prepared. So the function of these follicular stimulating hormones in male is that it stimulates and acts on the sertoli cells. As I have already discussed, have given a gist about the sertoli cells, right? So now you can understand very well. So this follicular stimulating hormone will stimulate these sertoli cells or acts upon these cells. As a result of that, these sertoli cells will have positive effect on spermatogenesis. Whereas what does this LH hormone has, what is the role of LH hormone? As we know, luteinizing hormone acts on the Leydig cells. We already know Leydig cells are the interstitial cells. They are not present in the seminiferous tubules, but they are formed outside the seminiferous tubules in the interstitial space that is present among and between the foraminiferous tubule. So these Leydig cells will, in response to this stimulation, releases the androgens, and the special type of androgen is testosterone, and this testosterone has many effects that are called androgenic effect. So this hormone will control the secondary sexual characters, as well as it also has a role in it also has effects on spermatogenesis. Whereas what is the role of Sertoli cells? What is the role of Sertoli cells? These Sertoli cells has positive role on spermatogenesis, as well as they will release one type of one inhibin, one substance known as inhibin. This inhibin has negative feedback regulation. By acting negative feedback regulations by inhibiting the anterior pituitary. So this is how the regulation is also known. Apart from this, Sertoli cells also release androgen binding protein. They also release androgen binding protein. Apart from this, the Sertoli cells also has a nutritive function, and so the Sertoli cells has different names. Let us go and study here. So here you can appreciate very well that here are the Sertoli cells that are lying here only. At the same time, the Leydig cells are here. Leydig cells are also known as we know, interstitial cells of Leydig, and Sertoli cells are also known as nurse cells or supporting cells or nourishing cells or sustentacular cells. So there are different names for Sertoli cells because because of its function. Because Sertoli cells also helps in nourishment, they are also nourishing of the spermatozoa, the developing spermatozoa. So it is known as nourishing cells, supporting, as it is supporting as a nurse, it is working as a nurse, and sustentacular cells. So this is how you have to relate, and also you can remember these names along with their function. So after this, then the question arises, what is we are saying that it has a they will have a positive effect on the spermatogenesis, but what is that positive effect? That is very important. On which cells they act positively in further stages, so that this spermatogenesis is initiated? The answer is primordial germ cells. About what is primordial germ cells? So to understand what is primordial germ cells, so for that, let us move to our next slide. So my dear friends, here in this slide I'll be explaining totally about what is primordial germ cells. The what we have at least we have came to know that all these process are initiated spermatogenesis as the primordial germ cells. The only reason behind that is the primordial germ cells were activated, so that all these spermatogenesis is occurring in the body at the age of puberty. So let us understand how these primordial germ cells have formed embryologically. If you see the image here, this is the zygote, my dear friend. This is zygote. As we know, zygote is the product of fertilization. So let us move forward, and this zygote undergo many divisions and form blastocyst, and this blastocyst has inner cell mass, and in the inner cell mass it has epiblast. And what you have to remember here is that this epiblast is the thing what is leading to the what is the precursor of the primordial germ cells. These are the primordial germ cells, and this is the precursor. This is the precursor of the primordial germ cells. So epiblast is the embryologically precursor of the primordial germ cells, and these primordial germ cells are the main thing that is important for this all the cycle. And as soon as the age of puberty occurs, these primordial germ cells at the same time, these before that, these primordial germ cells have to, they are present in the yolk sac. They are present in the yolk sac. So in the embryonic development, they go here, they migrate from the embryo yolk sac to the genital ridges of the embryo, and in the genital ridges they undergo many divisions, and also they are the reason for the development of the development of the reproductive system in total. So I'll be dealing with that in detail in another video of embryology of reproductive system. Now you have to just remember that they are migrating to from the yolk sac to the genital ridges, and in the genital ridges they stay in the dormant they stay in the dormant stage. And as soon as the puberty as soon as the onset of the puberty, as a result of positive stimulation of the spermatogenesis, regulation of the spermatogenesis, these primordial germ cells will undergo differentiation, that is the cell divisions, and form the gonial cells, and then at last they form the primary gonocytes, what we known as, if it is in the male it is called the primary spermatocyte, in female primary oocyte. Now after these primary spermatocyte form, after that the meiosis occurs, and then the further metamorphosis, all those occur and lead to the formation of the gametes lead to the formation of the gamete by the process of gametogenesis. This is ovum and these are sperms, and these sperms again undergo fertilization and the process of formation of zygote and the blastocyst, process of formation of the fertilization, process of zygote, and then the blastocyst, and then the epiblast, and this is forming then from the blastocyst, the epiblast, then the primary germ cells, and this cycle is again going on. So the primordial germ cells, the importance of primordial germ cells you have to know, and you should also know that this is originating from the epiblast of the blastocyst, from the inner cell mass. So all these are very important. Now after understanding all these things, let us go and study some clinical points and assertion and reason type of question. My dear friends, here the assertion is as follows, that spermatogenesis occurs after puberty. As we all know, we have studies very in detail. So spermatogenesis occurs after puberty. Yeah, it is correct. And reason is saying that primordial germ cells remain dormant from 6th week till the 6th week of development till the puberty. The reason statement is also correct, and as we know that primordial germ cells are remaining dormant, that is the reason why this cycle is not initiated in the all throughout childhood till the puberty. So yes, the reason is correct and assertion is also correct, and as reason is the correct reason of the, and this is the correct reason for the assertion. So this is how you have to tackle the questions. So my dear friends, the next question is about the teratoma. Teratoma are also known as the germ cell tumors, and what is the characteristic feature of these germ cell tumors is that they have all the three germ layers, that is ectoderm, endoderm, and mesoderm. So we know that teratomas and these have, they are saying that yeah, of course this is right, assertion is correct, they have all three germ layers. And what can be the reason is the question. So they are formed as a result of aberrant migration of primary germ cells. So is this correct? Of course, primordial germ cells. So the reason is also correct, and primordial germ cells are pluripotent. So as these primordial germ cells are pluripotent, they can form all three germ layers, all three germ layers. So reason is also correct. These teratomas are formed as a result of aberrant migration of the primordial germ cells, and primordial germs are pluripotent cells. That is the reason why these teratomas also have all the three germ layers. So with this we end our class today. So in next classes we'll be dealing with other with another good another topic of reproduction. Keep supporting. Thank you one and all. Goodbye.